Procedure of gram staining reaction. Addition of primary stain. Gently flood the slide with crystal violet solution for up to 1 minute. Gently wash with tap water to remove the excess stain and drain. Addition of mordant. Gently flood slide with grams iodine solution for 1 minute. Then gently wash with tap water. Decolorization. Add the decolorizer, that is ethyl alcohol, dropwise until the violet color is no longer emitted from the smear. Decolorization is followed by washing with tap water and drain the slide. Addition of counter stain. Flood slide with safranin solution for up to 1 minute. Then wash off with tap